I'd like to introduce Kattie Weigh, who's presenting today's webinar on the influence of soil pH on microbial community structure and function. Kattie completed a PhD earlier this year in soil microbial ecology at the University of Queensland. She's passionate about better understanding how microbial community dynamics regulate soil and ecosystem health to ensure the provision of ecosystem services for future generations. She's got a strong skill set in bioinformatics, statistics and soil DNA methodologies. Kattie has been a part of the New South Wales DPI soils research team for not quite a year yet, working on a number of projects including defining microbial indicators of soil health, building resistance and resilience in soils, and exploring the microbial remediation of hard-setting soils.
Okay, thanks Luke. Thanks for the introduction. So I've been with the DPI soils team at Wollongbar for about 5 months now. As Luke said, I finished my PhD in January, so I'll be presenting some findings from my PhD project. Firstly, I'll provide a bit of context and key discoveries that inform my PhD project. Then I'll present my work where I characterise the influence of soil pH on bacterial and fungal abundance and diversity, and present an example of how pH regulates microbial community function. So, soils host a quarter of our planet's biodiversity. Just a single gram of soil can harbour 10 billion microbial cells, which means there are more living organisms in a teaspoon of soil than there are people on the earth. On top of that, every species has a unique genome with thousands of specific genes. Soil microbes mediate a wide range of ecosystem services that humanity depends on, including nutrient cycling, carbon sequestration, and the provision of food, fibre, and fuel, and many more. Microbes inhabit virtually every environment on Earth, from rainforests, grassland, and oceans, to the human body, to pretty extreme environments such as hot springs, frozen tundras, volcanoes, and acidic lakes. Okay, so microbes are everywhere, but how do we study their spatial distribution patterns? Spatial distribution patterns of macroorganisms have been studied for centuries, but when it comes to microbes, it's a bit more difficult. About only 1% of microbes are amenable to lab-induced growth conditions. The development of culture-independent methods, such as DNA sequencing over the past 2 decades, has not only enabled Earth's microbial diversity to be characterised, but has enabled sampling and profiling of whole communities. This has meant that investigating spatial patterns of microbes has only been possible more recently. So yeah, biogeographic surveys of microbes in soil environments have identified a range of variables that regulate the diversity and composition of microbial communities, including soil nutrients, such as carbon, nitrogen, and phosphorus, chemical properties such as pH, and above ground vegetation. So there's a whole bunch of environmental variables that regulate the diversity and composition of microbial communities. But a study of 98 soils across North and South America actually found pH to be the strongest predictor of bacterial phylotype diversity, as we can see here, and richness. So we've got pH on the x-axis, the diversity on the y in the first figure, and the one below it, richness, on the y-axis. Interestingly, soil pH was a much better predictor of diversity than geographic location, ecosystem type, and large climatic variables, such as temperature and latitude. For those who don't look at these sort of figures very often, these two in the right panel, the samples are basically plotted in multi-dimensional space based on their similarities. In this case, soils harbouring more similar microbial communities are positioned closer together. So looking at the shapes in both of the figures, we can see samples are distinctly separated by pH rather than the biome type. A later study profiling a wider range of terrestrial biomes also revealed an increase in bacterial phylotype diversity and richness towards more neutral pH, and again found the compositional distribution of microbial communities to be predicted by pH rather than ecosystem type. Because of this discovery, researchers then wanted to isolate the effect of pH on soil microbial community dynamics. And the Hoosfield acid strip is a perfect place to do this. So the Hoosfield acid strip runs alongside an experimental farm that has supported the growth of barley for about a century. In the late 1800s, biological limestone was excavated from nearby Bell Pit mines and carried across the site in wheelbarrows, meaning the lime was applied differentially across the site ranging from about 150 to 250 tonnes per hectare. Chalk reserves have been subject to biogeochemical cycling and leaching since, forming a gradient of calcium carbonate where soils increase in acidity with distance from the chalk pits. With a pH range of 4 to 8 within about 200 metres, this silty clay loam can be analysed without variation in potentially confounding large-scale variables such as latitude and temperature. The Hoosfield acid strip is now a uniform pH gradient that has served as a novel substrate for experimentation since the 1950s. So within the range of about pH 4 to 8, Rusk et al. Found that the number of bacterial taxa doubled within this range, and community composition differed significantly, showing strong discrimination with pH. The numbers of fungal taxa did not significantly differ with pH, and while this association between pH and fungal community composition was significant, the ordination did not reveal really clear discrimination with each pH unit. So if pH has such a huge influence on microbial community structures, then what influences pH? So obviously there's a whole range of abiotic, biotic, natural and anthropogenic influences on pH, such as land clearing and land use change, fertilizer application, and things like that. So if we delve into a bit of the research I did in my PhD, I actually also studied the acid strip in Rothamsted that I just showed results from. My supervisor, Paul Dennis, he did his PhD at Rothamsted, and this is the stretch that was sampled. So at each half pH increment, 20 soil cores were collected. When we looked at the pH of those soil samples, we found that pH ranged from about 4 to 8 within only about 70 metres. So the solid points on this plot were the ones that were included in my PhD project. I removed these hollow samples as I already had full coverage of the pH range at this site. So firstly, DNA was extracted from each of the soil samples, and the genes conserved to bacteria and fungi, namely bacterial 16S and fungal ITS, were quantified using real-time PCR to infer bacterial and fungal abundances. So within the range of pH 4 to 8, bacterial abundances, as inferred by gene copy number, increased linearly with pH, and fungal abundances peaked between about pH 5 and 6. Phylogenetic marker gene sequencing was then used to profile microbes that inhabit the samples along the gradient. In line with the previous findings from the site, I found that the observed number of bacterial taxa increased with pH, and as did the diversity of bacterial taxa. And bacterial community composition shifted significantly along the gradient, with soil pH explaining about 69% of the observed variation in samples. So this figure looks a bit overwhelming to start with, so I'll just quickly break it down. Each column represents a different soil sample, and they've been ordered by their pH value, which is shown by the bar at the top. Each row represents a different OTU or taxa, and they've been organised by their phylum class order sort of blocks. The shading denotes the abundance of that specific taxa within a specific sample, where white means it wasn't observed at all and darker colour means it was highly abundant in that sample. So we can see that individual bacterial taxa showed strong responses to pH, with the Acetobacteriota phylum showing a pretty split response along the gradient, some favouring acidic conditions and others preferring neutral to slightly alkaline conditions. Bacteroidota, this phylum here, this block, we can see that there was a notable increase from slightly acidic to neutral pH, and the vast majority of Chloroflexi preferred acidic conditions. These really smooth transitions in abundances suggest the presence of 3 ecological groups within the pH range of 4 to 8. Because these transitions were observed and I could see sort of 3 potential groups in the data, samples were then hierarchically clustered based on similarities in their taxonomic profiles. So here, rather than ordering samples by pH, as I did in the previous slide, here they have been clustered by community similarity. We can see that the samples fall into 3 groups, and if we look at the pH of each sample, we can see distinct groupings by pH. These results suggest the presence of 3 alternative ecological states defined by 2 tipping points. Okay, so now that I've explained the structures of the sorts of figures I'm presenting, in the essence of time, I might just move through the fungal results a little faster. So fungal richness and diversity increased with pH, and fungal composition differed significantly with pH. The individual fungal taxa showed strong responses to pH as well. The Ascomycota phylum showed pretty split responses along the gradient. And while the 3 bands are less obvious here than the bacterial heat map, the suggestion of 3 ecological groups remains. And when we look at the hierarchical clustering of samples, yet again, 3 groups is observed. So we know that soil pH influences bacterial and fungal biomass and diversity. We also know pH drives major shifts in community composition, potentially suggesting the presence of 3 distinct communities within the range of pH 4 to 8. And as you'll recall, microbes like fungi and bacteria form important ecosystem services that we rely on. So imagine if we had 2 samples from a site, one with a pH 7 that falls in this group, and one with, say, a pH 5 falling in this group. For whatever reason, say one was managed and one wasn't. Based on the results presented here, we could expect the microbial communities of these 2 soils to be quite different. The question then is, if the communities are significantly different, will the 2 soils uphold the same ecological processes? To delve into this question, I looked beyond taxonomy of these communities and explored their functional traits. So rather than sequencing a single gene of interest, as we did before with the phylogenetic marker gene sequencing, instead, all of the DNA was fragmented and sequenced, which allows the entire genomic content of the samples to be realized. Here I applied this method to study genes involved in the nitrogen cycle. Bacterial genes are presented in rows, and again, the columns are each individual sample ordered by their pH value. And each gene is organized within its overarching metabolic pathway. Asterisks indicate whether the genes were significantly associated with soil pH. And yet again, we do see the suggestion of 3 groups in response to pH, but this time with gene abundances rather than taxa. And if we look at hierarchical sample clustering, yes, we see 3 groups is confirmed. So this suggests that soil pH does not only drive compositional shifts in microbial communities, but informs distinct zones of functionality. These analyses also provide insight into which metabolic pathways are expected to strengthen or weaken under different conditions, and how that might contribute to key processes in the nitrogen cycle, such as nitrogen gain versus loss from a system. Here I've sort of mapped out the global nitrogen cycle. Just to break it down a bit, all of the different nitrogen compounds are in these blue tiles. All of the arrows and the colors of those represent different metabolic pathways. And these little purple tiles indicate the genes involved in those metabolic pathways. And then pretty small next to those tiles is a pretty basic plot. Basically, the x-axis is pH and the y-axis is the gene abundance within each sample along by pH. I've just fitted a linear curve to all of them. This figure could be improved, but it just sort of gives you a general idea about how you can sort of map how different functions in the nitrogen cycle may increase or decrease or create an imbalance along the pH gradient. So anything that increases gene abundances that increase with pH have a blue model fitted, and those that decrease with pH are red. Those are neutral, are black line. So we can see, for example, that the relative abundances of genes involved in ammonium assimilation increase with acidity, while those involved in denitrification generally increase towards neutral pH. So yeah, we could end up with an imbalance in the nitrogen cycle if pH was to change. So I guess the take-home points from the research that I presented here is that soil pH affects microbial community biomass, diversity, composition, and function. It also drives distinct patterns of phylogenetic and functional diversity, where pH 4.5 and 6.5 resemble potential tipping points for alternative ecological states. So ultimately, it's important to manage soil pH to ensure the provision of microbially mediated ecosystem services that we rely on. And I'd just like to acknowledge the Rothamsted Research Institute and the University of Queensland, as well as my principal advisor, Paul Dennis, and my co-supervisors, Suzanne Schmidt and Robert Halsey, and my beautiful lab mates at UQ. Thank you.
I was wondering, why do you think it is that the pH had more of an outstanding effect on the bacteria than the fungi?
Thanks, Blake. Bacteria typically inhabit more narrow pH ranges, where, say, a small incremental pH change can really affect their growth. Fungi, on the other hand, can span about 5 to 9 units of pH without it even impeding their growth. So on that note, if an organism is living within their optimal pH range, other soil factors like soil carbon content may actually exert a greater effect on the abundance than pH. But as soon as the pH deviates away from this optimum range, it appears that pH exerts the largest influence.
pH 4.5 and 6.5 being tipping points, can you comment on how long it takes for microbial communities to respond to treatments to shift pH, say in response to lime applications in cropping or in pasture paddocks? Great question, Mary.
Yeah, that is a really good question. I guess that sort of comes down to the resistance and the resilience of the soil. So resistance being, if pH is to change, how long can your community keep performing those functions in a suboptimal, I guess, chemical environment? And then resilience being, say if the community does shift, because obviously, once pH changes, it may not be immediate. They may be resistant for a certain amount of time. And then once the pH does change below one of those tipping points, then I guess it's the time, the soil is amended. Then I guess now we're thinking about the time it takes for that soil to recover back to its original state. So I imagine that this would vary soil to soil based on, how resilient the soil is, potentially how diverse the soil is as well. If you have a lot of functional redundancy in a soil, if some of those organisms or their genes get knocked out, if there's a reservoir of other genes that can perform those functions, then that functionality might come back sooner. But yeah, I would just say it's down to the resilience of the individual soil. But really good question. I actually just had a follow-up point. I haven't included the results in this presentation, but another thing I did find along the pH gradient, when I looked at the structures of the genomes along the gradient, I actually found that a lot of the genomes actually reduce their size. It's a phenomenon called genome streamlining. So when microbes are stressed, responsive to all different types of stress in extreme environments or along gradients, they actually discard any genes that are superfluous or not serving them so that they can sort of optimize their energy usage to survive. So that may be why they have greater carbon use efficiency, like that was mentioned, at the more acidic end of the pH spectrum.
So, Kattie, you used shotgun metagenomics to look at the abundances of the genes that were involved in that nitrogen cycling. Do you reckon this method can be applied to other genes and functions?
Thanks, Blake. Absolutely. Basically, using this sort of a method where we extract DNA, chop up all the fragments, and then we sequence the whole metagenome of it. Once you have that metagenomic data set, you can really screen for any genes. We're pretty much only limited to how comprehensive the databases of genes are. We can also use a method to characterize gene diversity. So rather than just the abundances of genes that I showed here, we can actually look at the diversity contained in different samples, which will provide information about, say, the functional redundancy of the soil, which is important in understanding the resilience of the soil. For example, with greater diversity of genes that are performing the same function, if some genes are lost upon disturbance or stress, like I just mentioned with the genome streamlining and things like that, if we have a reservoir of alternative genes, what we call functional redundancy, that can be really important for the, I guess, sustained provision of that function.
Cool.